It is my pleasure to introduce Dr. Fan Lam, who is currently at the University of Illinois Urbana-Champaign. He's an assistant professor in the Department of Bioengineering and faculty at the Beckman Institute. He has been doing work focused on magnetic resonance-based molecular imaging and multimodal brain mapping. And he's been studying, brain function, both in normal and disease states. So he has been, doing a lot to contribute to the field, and we're looking forward to seeing what he's going to be talking about today, focused on spectroscopic imaging. With that, I will let you take it away, Dr. Lam.
Thank you so much for the kind introduction. Yeah, and nice to meet everyone. It's really great to be here and to see everyone online. Just like to let you know that I have a little bit sore throat going on, so this is not what I sound normally. So next time if we meet each other in person, don't be surprised. I'm still me. There's no spam or anything. Okay. Yeah.
You can just blame it on WebEx.
Yeah. Yeah, very happy to be here today, and I would like to share with you some of our efforts on developing advanced new imaging technology to provide molecular level information in the brain, and particularly on this front of trying to achieve more quantitative multidimensional MR spectroscopy imaging. So this probably compared to fMRI, diffusion, and other more anatomical imaging strategies, this might be a little bit less familiar, but I hope throughout my talk, I will convey some of the importance and the promise of this very exciting technology to allow us to study the brain at the molecular level and perhaps connect to the functional level of measurement. Yep, let me try to see if I can move this forward. All right, yeah, I don't think I need to preach to this community on the significant role that MR-based neuroimaging techniques have been playing and in the study of the human brain by allowing us to map very detailed structural functional organization of the brain and to extract some quantitative measure in different brain regions and even allow us to quantify tissue properties that potentially provide different level of biomarkers about the brain functions and disease. And this has been really transformed the study of human brain in vivo for the past 2 to 3 decades. However, one thing that we need to recognize here is that all this amazing information that we have been using and benefit a lot came from a single type of molecules alone. That is the water molecule in our body. And particularly, they are all generated by signal coming from the hydrogen atoms on the water molecule. And then in the meantime, it's been increasingly recognized that in each microscopic voxel that we are measuring in the MRI experiment, the tremendous richness of the molecular level complexity in the brain, and so many different molecules in there, ranging from small metabolites, neurotransmitters, lipids, peptides, proteins, and even DNA, and the dynamics. So, and they all play different physiological functions, very important physiological functions, and understanding their relative abundance and their dynamics, their interactions, have a lot of implications in terms of the basic science study of a physiological process such as neurotransmission, energy metabolism, and then some pathological processes related to many different neurological disease and neurodegenerative disorder, and try to understand this molecular level complexity will potentially provide a new dimension of information to allow us not only better understand the molecular level and molecular underpinning of different brain functions, and also provides potentially new biomarkers for different disease clinical applications, ranging from diagnosis, prognosis, and also offer new tools to monitor treatment efficacy, etc. So in the meantime, we have to recognize that compared to the development, the amazing development in the functional imaging side for the past 2 decades also, the tools to measure, to resolve this molecular level complexity in the brain and in vivo particularly have been lacking. So our group is driven by this motivation and trying to fill the technological and knowledge gap by developing new technologies and focusing on the following fronts. First, trying to develop new techniques to allow higher resolution, more quantitative, multiplex molecular imaging of the brain. And secondly, leveraging the recent advance in machine learning and deep learning to integrate physics-based modeling and data-driven machine learning strategies to develop new techniques that allow us to enhance the data processing and image reconstruction and quantification process for higher dimensional MRSI data, which I will get into in more details later. And then another theme that we have been recently venturing into is to provide new tools to allow multi-scale, multi-model biochemical characterization of the brain. So in today's talk, I will try to focus on the two themes that highlighted here. So before I go into the details of our work, so I would like to introduce some background information of MR spectroscopy imaging, aka MRSI. Since this may not be a modality that well known to most of the audience here. And so you probably know that in MRI, the sensory data we collected is in the data domain, which is not exactly image domain, which is called the K-space. And so in MRSI, instead of scanning through the K-space here, we are also collecting for each K-space point a free induction decay shown on the middle here in the grayscale here. And then after we have this high dimensional data, basically we have this additional encoding dimension associated with each K-space point. After this, we can do some kind of Fourier reconstruction and to convert this KT space data into some kind of XF space data, where you will have spatially resolved spectral information. It's kind of combine the power of MRI, spatial encoding of MRI, and then the chemical specificity offered by chemistry and NMR spectroscopy to allow us to obtain spatially resolved metabolic or biochemical information. For example, if you look at this spectrum here, you have different peaks corresponding to different molecules. And then by quantifying them, you can study their relative composition and providing a very rich set of molecular level biomarkers. And the more amazing thing is that you don't necessarily have to be restricted to the proton nuclei. You can also try to detect signal from phosphorus nuclei to study energy metabolism, or you can detect signal from carbon-13 containing molecules, and potentially offering a quite comprehensive characterization of the metabolic profile in a spatially resolved fashion. And if you do proton and phosphorus, you can do it in label-free. So this potentially provides a very powerful molecular imaging, multiplex molecular imaging tool for study the brain in vivo. So here's another kind of motivating example I would like to give. In this case, this is a study published by a group from UCSF, leading group in the study of brain cancer using MR spectroscopy and spectroscopy imaging. So in this case, this is a tumor patient. If you see that this typical clinical scan, you will see in the T1 contrast enhanced scan, this is a small tumor highlighted here, small lesion highlighted here. And then they also did a PET scan, see some kind of a little bit abnormal, a spreading abnormality in the brain as well, beyond what is visualized in the T1 contrast enhanced image. And then they did a spectroscopy scan, allowing to get not just a single molecule distribution, but this kind of molecular profile information across different locations of the brain. And they identify in the spreading region, you do see an elevated choline to NAA ratio, which indicated that this is actually a tumor region spread far beyond what was highlighted in the T1 gadolinium enhanced image. And this was later confirmed by surgery. So this kind of early work really highlight the potential of having this multiplex molecular level information in helping us better to diagnose and design better treatment or patient management strategies for different clinical populations. So with all this promise, one of the major issue with developing MRSI for in vivo application is that the development of these things have been progressing slower than expected. So here are some of the fundamental technical challenges that limit the development and widespread use of MRSI, particularly as you will see that we're not only acquiring a spatial image, like 2 or 3 dimension, we are also adding additional dimension to the original imaging problem, making this a higher dimension problem. And you need to encode the spectral dimension for every voxels in the brain that you're trying to image, and making this a very high dimensional and slow imaging modality. And more importantly, this molecular level information, although very important and powerful, the concentration of this molecule we're trying to image in MRSI typically has a much lower abundance compared to water molecule, typically 3 orders of magnitude lower. So it's intrinsically a very sensitivity limited imaging modality. And on top of the high dimensionality and sensitivity challenge, we are also facing a tremendous challenge from accurately separating different molecular components. As you will see in this one-dimensional spectrum that we acquire from an actual brain, you can see that the spectral features come from different molecules. They kind of overlap together. So except some major metabolite like NAA, creatine, choline, that was much easier to measure. But if you're interested in neurotransmitter signals such as glutamate and GABA, which is very important for brain function studies, you can see that the overlapping spectrum really prevents us to accurately separate them and provide more robust quantitative measurement. So in the to address all these challenges in the past decades, many methods has been proposed to improve MRSI, ranging from fast sequences, trying to acquire data as fast as possible, and many different constraint reconstruction methods try to incorporate different prior information to improve the reconstruction of the high dimensional spatial spectral function from limited or noisy KT space data, and also the development of advanced instrumentation going from 1.5 Tesla to 3 Tesla to even 7 Tesla and higher, and also phase array coil for surface coil to improve the sensitivity of the detection. So addressing these issues in a component-wise fashion, although have led to very exciting improvement, but the reality is that if you go to a scanner, if you want to acquire spectroscopy scan, most people still do single voxel spectroscopy without any spatially resolved information. The best spatial information is where you place the voxel. And if you want to do an imaging scan, you typically confine to this level of performance with 1 centimeter cube size voxels, long scanning time, and typically you can do 2D because 3D scan is too slow. So to address these fundamental technical challenges, we really try to think about how to tackle these things from end to end. So redesign the acquisition, processing, and quantification strategy to offer a different kind of way to perform MRSI. So working with my previous PhD advisor, Professor Jipeng Liang, also from UIUC, we developed this subspace imaging approach called SPICE to address this dimensionality challenge and then sensitivity challenges. So the underlying framework of SPICE, the mathematical framework of SPICE, is subspace modeling. So instead of treating each voxel and each spectral point as independent measurement that we have to acquire in a very high dimensional space, we sort of recognize that if you look at each voxels, the spectral composition only comes from a finite number of molecules that can be detected by NMR in vivo. So this is inherent dimensionality, very low dimensionality in the data that embedded in this very high dimensional space. So driven by that, we can sort of express this high dimensional function at each voxel as a linear combination of a small set of basis functions. And if you can predetermine the spectral basis function, if I will talk about more later, then you convert this high dimensional imaging problem into a much lower dimensional imaging problem where you only need to recover a set of low dimensional spatial coefficients. And then you can compare the number of degree of freedom reduction ranging from number of voxels to number of FID points, which can typically be very, very large in order to achieve a high spectral resolution. And that will be reduced to the number of voxels times a very low model order, which can be orders of magnitude smaller than the number of voxels and number of points. So this significantly reduced dimensionality allow us to rethink about how we will acquire the data and process data to break out the traditional SNR speed resolution trade-off faced by the conventional Fourier imaging and paradigm. So using the SPICE framework, I'm showing you some results from earlier. We are able to achieve almost whole brain fast, high resolution MRSI of the brain, and close to the level of spatial resolution that achieved by functional fMRI, but with only less than 10 minutes. So this is order of magnitude faster than if you have acquired this level of resolution data using, say, a chemical shift imaging standard CSI sequence that's available in a clinical scanner. We also have some preliminary translational effort with this early phase of SPICE development, and we can image brain tumor patient with very, very high resolution. In this case, we can achieve, again, isotropic 3 mm resolution in less than 7 minutes. And this is in collaboration with Zhejiang University. And we show that this high resolution capability really help us highlight much better the tumor region identified by this elevated choline to NAA ratio. And if you will acquire this level of resolution in clinical, it will take you hours to, using the clinical scan, it will take you hours to generate such a data. And this is more recent data where we apply the subspace imaging processing to existing sequence in collaboration with Julie Pan from University of Missouri. And in this case, we're not trying to go to 3D, and we're trying to achieve, let's say, can we do a 2D scan as far as possible to minimize, the scan, additional scan time that we add to a tumor imaging scan without complicating the imaging protocol. So in about 2-minute scan, the data is not super high resolution, but as you can see, compare the data produced by our subspace-based processing and the standard Fourier reconstruction, we can achieve better SNR and better reviewing different molecular features. And if you look at the choline to NA ratio, it better highlight the tumor region. And this was confirmed to be a tumor recurrence case with our collaborator. So really exciting result to showcase that the molecular level information can better determine this tumor, whether it's a treatment, the abnormality we observe in the anatomical scan, whether it's due to treatment effect or actually tumor recurrence. So all this exciting capability, high resolution, fast MRSI enabled by the SPICE framework really motivate us to think about, can we push this even further to provide molecular level information with more quantitative accuracy and robustness? And then also, can we get more information from the spins at different molecules? So this is the drive behind our effort on developing this new set of imaging techniques called multidimensional MRSI for high resolution quantitative molecular imaging of the brain. So to talk a little bit about why we want to do it, right? So we already can do very high resolution MRSI. So why we want to add additional dimension to the imaging problem? So if you look at this MRSI volume, you get a voxel, you get a spectrum from every macroscopic voxel. So very high quality. But the problem is that if you only get a single frequency spectrum with a single frequency dimension, and you look at the spectrum, it's really, really crowded. And you have signal coming from the major metabolite that people have been used to study cancers for years, and acetylcholine, myoinositol. But there's also a very interesting component coming from neurotransmitter pool like GABA, glutamate, and others. There's also macromolecule component, which can affect the quantitative accuracy of the metabolite, but also itself could serve as additional biomarker in different disease. So if you look at this single spectrum, it's very difficult to separate different signal. In the signal processing term, this problem is very yield condition to accurately separate different molecular components because they are overlapping spectral features. And also very sensitive to noise. And then you can also look at this large dynamic range from different molecules, right? Even though they are all low concentration, but compared to NAA, the signal of GABA and glutamate is much, much weaker. So separating them is very challenging. So this is one reason we want to see if we can better separate this. And the other benefit of introducing additional dimension is that you can actually do a lot of things to different molecules that you have been doing for years for the water molecule. For example, you can introduce T2 weighting to the water to the spectroscopy acquisition. You can introduce diffusion weighting to the MRSI acquisition. So this not only allows us to better separate different molecules by exploring their property on this additional dimension, but also allow us to quantify molecule-specific biophysical parameters, such as T2 relaxation, T1 relaxation, and even diffusion properties. So what we want to get is biophysical property beyond just relative abundance of different molecules. Well, I think the power of, let's say, functional MRI and diffusion MRI already speak to why we want to go beyond just proton density. And in the meantime, they do provide additional information. Like, for example, in this case, it was a stroke study to try to track the recovery of ischemic stroke patient. And they do single voxel spectroscopy, of course. And if you look at the ADC of the water molecule, it gives you this very consistent data that kind of consistent with what we have been understanding about stroke, right? ADC keep increasing during the recovery of the ischemic stroke. But if you look at not just the molecular concentration itself, but also the T2, for example, constant of the NAA measured by the single voxel experiment, you can see this T2 of the NAA molecule, which reflect a different microstructural property of the tissue, show a very distinct and interesting pattern compared to the stroke, compared to the water information. And this all speaks to the potential of that if we not only can extract concentration, but also some biophysical property of the tissue, molecular specific biophysical property of the tissue, we could potentially provide much richer set of biomarker. If single voxel can do this, imagine what imaging, high resolution imaging could give you, right? So the problem to do this is that very challenging. So this diagram shows what we will do in the conventional single TE MRSI experiment. So basically you will get a single spectrum out from every pixel, every voxel in the brain. So in order to encode additional dimension, for example, you can encode the TE dimension. So what you have to do is to collect this data at different TEs and introduce different relaxation weighting. So you introduce so you can encode additional property about different molecules. But the problem is that you add another dimension into an already high dimensional imaging problem, which has more limited trade-off between speed, resolution, SNR, and there's also SNR efficiency consideration given the traditional spin echo acquisition, because you need a longer TR for the spin to recover. So you are not effectively leveraging the longitudinal recovery period to collect the data that you want. So to address this issue, we try to develop an end-to-end strategy, trying to rethink about how we will acquire the data and process the data in an integrated fashion. So this is our kind of one of our current acquisition strategy. So we are extending this to a multi-band acquisition, which allow us to cover a big area, almost whole brain area, in the same time as you will acquire a single volume, 3D volume. And you can excite this slab sequentially within a TR to maximize the acquisition efficiency within the issue repetition time, which people have been doing for SMS imaging in functional MRI. And in each slab, we're combining some selective refocusing strategy and then the rapid spatial spectral encoding. And I'm not going into the detail of this, but basically trying to maximize the acquisition efficiency, like maximize the number of data and then the SNR per unit time that you can get. And also, we also try to leverage this longitudinal recovery time, which during when in the conventional acquisition, you're doing nothing, but we try to acquire some additional data to leverage this free period. For example, we can interleave some water acquisition and acquire some interesting data that you will have done using water imaging. So using this, you can extract not just MRI image information, water image information, but also some system information that can be useful for data processing. So eliminate need of additional calibration scan to further reduce the total acquisition time. So such an acquisition really generate very unique data set that you couldn't be handled by conventional processing, particularly Fourier reconstruction method. And so the way we handle it is we develop this union of subspace that really extend and build on the original SPICE framework. In this case, we are developing this augmented subspace model where we further represent this very high dimensional spatially resolved temporal data with additional dimension, denoted as T1 here, into different components. And we assume each component have reside in their own individual subspaces. And one example here is that we can further decompose this high dimensional function, multidimensional function, into a major metabolite component capturing mostly things that you will do using single T MRSI, but also additional neurotransmitter components such as glutamate and GABA. So, and you assume each one of them reside in their own low dimensional subspace. This not only significantly reduce the dimensionality degree of freedom for the imaging problem, but also provide you a way to better separating them by exploiting their unique subspace structure due to their molecular property and the acquisition design that we have. So one of the key questions now here is that to make this work, you need a way to actually learn these subspaces instead of jointly learn them from the high resolution, which is very noisy data. And how do we introduce the years of knowledge of chemistry study, biochemistry study, and NMR spectroscopy study into this approach to allow us to design a physics-driven subspace learning strategy to allow us to pre-learn this subspace and then transform this high-dimensional imaging problem into the problem only need to recover this low-dimensional spatial coefficient. So without going into too much detail, hopefully just convey the concept here in terms of integrating spin physics and machine learning. So we can actually predict in the ideal experiment what the resonance structure of all the molecules that we are interested in and can be detected in vivo should look like. So we can predict them using quantum mechanics. So you don't have to reimagine every experiment. And there's been years of study of well-established biophysical model that describing what a spectroscopy signal should look like mathematically. And if you look at this equation, there are some things that can be predicted using physics, such as the phi, which is the quantum mechanical basis here. And then there are some small number of molecular dependent parameters like T2 concentration. And you can actually learn the distribution for these parameters using both experimental acquired data, specially designed high SNR data, as well as you can pull in number from literatures and trying to construct some kind of distribution of this parameter. So once you have these two elements, you can actually combine them and push them through the biophysical model to generate as many training data as you would like. And then you can kind of cascade them into some kind of Sarkasorati matrix where you can derive the multi-TE multi-dimensional basis function for different components using matrix factorization analysis. So, and the next question, once we have this subspace, do we just go through and do the experiment? How do we choose the parameter that we try to encode the data, particularly if we try to do multi-T MRSI to better separate neurotransmitter and metabolite? How should we choose the TE? Should we choose the TE that people have been used for years, like uniformly spacing many TE and acquire as many of them as possible, which is more a straightforward way to do this? Or is it possible to only acquire a selective set of echo time, which can be optimized for a particular task that you are trying to solve? In this particular case, the answer is the latter. So if you are interested in, for example, the best separation between metabolite and neurotransmitter component, say for glutamate and GABA, the answer is you don't necessarily have to acquire as many T as you like. If you do an equivalent time comparison, for example, you acquire many T versus less averages, or less T with more averages, and also an optimized selection set of TEs, you will notice that actually, if you want to separate glutamate and GABA in the equivalent setting, you should go for some optimal TE choices, not a uniform TE choices. Okay, and you can, based on the subspace framework and the physics model we have, you can tailor this optimization for different kind of acquisition that you want, and then optimize it for different component, become a very task-driven optimization. So here is some simulation to back up the theoretical prediction. So if your task is to maximize the performance of neurotransmitter and metabolite separation, as you will see here, this is a Monte Carlo simulation comparing different choice of TEs. And then you can see that compared to doing a very, very short TE, more averages, as people would have done conventionally, and versus a two shorter TEs and random 2 TE choices and optimized 2 TE choices. The optimal TE combination actually give you the best estimation performance in terms of minimizing the estimation variance. Again, you can do this for a task-specific fashion, right? You can also optimize acquisition for T2 mapping to map the T2 of different metabolite. Is it necessary that more T2, more TE helps you to achieve better performance? The answer is actually no. So if you look at this CRLB Kramer-Rao-Loebaugh analysis, which minimize the T2 estimation of NAA, creatine, and choline, the optimal number of T that you should choose in an equivalent time fashion is actually 4. So if you choose the T smartly, this analysis basically says that 4 Ts are sufficient for you to map the T2 of NAA, creatine, and choline reliably, and you can selectively choose those 4 Te values. So here are some comparison. So the 3 columns basically are results coming from different Te choices as well as the different quantification strategy. So the first 2 columns shows the result from a standard parametric fitting model, where you use typically 4 Te choices that people come out with, more uniform Te choices, and then the optimal 4 Te that we identify. You can see that using the same quantification strategy, you achieve much better estimation variance. And then if you further use our proposed estimation strategy, which kind of combining the parametric fitting model and the augmented subspace separation, we can achieve even better quantification performance. So that's kind of confirming the theoretical prediction. And we also try to develop this on in vivo, trying to acquire data based on this experimental design. So here shows some experimental result from a healthy volunteer. And this is a 14-minute scan. You can achieve 3.4 by 3.4 in-plane resolution. And then you can produce very high quality spatially resolved multi-T spectrum, clearly showing the T2 decay and the J coupling evolution. And if you really optimize this TE parameter, as we analyze, you can optimize it for this particular task of simultaneous metabolite neurotransmitter mapping. Here shows the mapping result coming from different 2 different subjects, showing that we can actually map the metabolite as well as the neurotransmitter component pretty nicely with very high resolution. Again, you can also optimize your acquisition for T2 mapping. So this is the high resolution T2 mapping that that have ever been produced, I think, in the field, like at this particular resolution, then we can really map out the T2 value of different metabolite in different region. Of course, technologically, there are still many optimization that we could do, better processing, better sequence, better artifact control to give us better and better result. But you can see the promise of this if you compare to the second row of the result, which people would have done using the typical TE choices, and compared to our choice of TE, you do see much better estimation. And you can do some regional statistics. You can see that the estimation variance is significantly reduced for our optimized TE combination. And we have collaborating with our local clinical collaborators. We're also trying to translate this to some clinical population, particularly with the ability to map more molecules more reliably, not just metabolite, but also neurotransmitter component. We are looking at this population called post-traumatic epilepsy. In this case, because the patient cannot tolerate a very long scan, so we try to shorten the acquisition, sacrificing resolution a little bit, but still significantly higher than you would have gotten in most typical scan. And in this case, in a 9-minute each scan, you can see that the high resolution metabolite and neurotransmitter maps. So I'd like to point out a few interesting observations here. And spatially resolved spectra are shown on the right with a symbol marked by the dot in the image. So you see two lesions in the flare image. This kind of dark space, which is due to the past injury, but probably acquired at different time points. And then you see this hyperintensity regions surrounding the dark spot, right? So they are both quote unquote lesions. But if you only look at the clinical flare, they are both lesions. So you cannot really tell what are the fundamental differences between those two, particularly biochemically. So with this capability, what we are showing is that the two different lesions are actually different at a biochemical level. You can sort of see it in the very simple observation of the ratio of the creatine and choline. And you can also look at the neurotransmitter, particularly the relative abundance between glutamate component orange here and the GABA component black here, and then compare to the normal appearance tissue. There are really interesting differences, and there are also lesion-dependent differences. There are a lot of heterogeneity within a lesion and across different lesions. And one of those, or both of those, might be seizurogenic. So we are trying to follow up with EEG analysis, trying to really explore the potential of this and carefully validate this. But the initial result looks really exciting. And this is really enabled by this multiplex, multiparametric imaging capability. Here's another very interesting case. If you look at an anatomical scan, you really don't see a significant lesion per se. And I'm not showing the clinical flare here. I should update that. But if you look at the clinical flare, you also see some kind of minor hyperintensity region. But if you look at the metabolite map, you see molecule-dependent abnormality in the molecular distribution, particularly highlighted by the arrow here. And this is where the some kind of hyperintensity you could identify in the clinical flare. And you see a significant reduction of NAA level, but you don't see that in the creatine, not so much in the choline. So if you look at the ratio between different molecules, you actually identify a very interesting abnormality that's not revealed by anatomical imaging. So a very interesting case. And just quickly, add that you can add additional dimension to the MRSI problem. You don't have to be constrained by adding, say, the TE dimension. You can also add different dimension. For example, in this case, you can add diffusion encoding dimension. My slides is okay. So skipping the detail of the sequence, you can basically combine different diffusion encoding module into our rapid acquisition module and sequence design, and you can encode the molecular metabolite specific diffusion process. Okay, and then you can leverage the similar concept by leveraging the diffusion model into this process to allow you to integrate biophysical prior into the subspace learning strategy, specifically designed for diffusion weighted MRSI problem. And here's some very interesting result. You can see that in this scan, we are able to obtain very high quality diffusion weighted spectrum from different spatially resolved voxel. And you can map metabolite specific diffusion coefficient with pretty high resolution for the very first time across the region of the brain. So this is, to me, this is really exciting. So this way go beyond a single voxel diffusion weighted spectroscopy measurement or just a diffusion measurement of the water, which is everywhere. But compared to water, these molecules are highly compartment specific and they are intracellular mostly. And achieve by probing, by the ability to probing their diffusion property could provide additional dimension of microstructure variation. So of course, this is still in the initial phase of development. We are trying to really push this from, but initial result looks very promising. We validate our sequence and against the conventionally used single voxel diffusion weighted spectroscopy. You can see the diffusion, the ADC measurement coming from our sequence is very consistent with the standard single voxel diffusion weighted spectroscopy scan. But we have to recognize that in our imaging scan, our individual voxel is 20 times smaller than what you have acquired in a single voxel experiment. We also did some preliminary evaluation on the in vivo case using, again, single voxel acquisition in a more gray matter rich area and a white matter rich area. And then the initial result also looked very exciting that we are producing consistent result compared to the single voxel diffusion weighted spectroscopy, but with much higher resolution. And then the difference between diffusion coefficient between different molecules are also pretty consistent with the existing literature. So just quickly summarize component that we're really pushing this from past high resolution multidimensional MRSI, and we thought that it's possible, and we can potentially robustly simultaneously map the metabolite neurotransmitter distribution in the brain. We can also extend this to map molecular specific biophysical properties such as T2, T1, diffusion, and potentially more. And we're trying to collaborate with different internal, external collaborators to explore the translational power of this new imaging capability. So pause here for like 1 minute and see if there's any question. I do see one question from the chat asking, do you think there's a future to do quantitative MRSI somehow? Yes, I think the short answer is yes. Thanks for the question. I think that's exactly what we are trying to do, trying to make MRSI robust and quantitative and provide as much multiplex information as possible, particularly multiparametric. Hopefully that answered the question. The other question is, how can you select the number of K-space? Yeah, that's a good question. So in terms of our current experimental design, we are not really optimizing the choice of K-space samples. We are more optimizing the encoding dimension along the additional parametric dimension, but certainly this could be done. But we are using some sparse sampling, leveraging parallel imaging capability and some sparse sampling strategy to allow us to extend the K-space coverage without significantly extending the acquisition time. Yeah. So I guess for the interest time, I would like to kind of finish the rest of the slides. And this kind of related to what I have been talking about. The second front is how can we better integrate physics-based modeling and data-driven machine learning for enhanced MRI processing and reconstruction. Some motivation behind this is that if you look at the generic spectroscopy signal model, it really can be decomposed into several components, right? Some component can be predicted by physics, some molecular dependent component that really captured by only a small number of parameters. And there are some experimental dependent distortion, which also reside in a low dimensional manifold. So the subspace assumption basically assuming that this signal, theoretically, they should reside in a low dimensional manifold because there are a finite number of parameters specifying their high dimensional variation. And if the variation of the parameter fall into a narrow specific physiological range for a particular population, The subspace model basically is an accurate approximation of the underlying manifold. But however, you do run into cases where subspace model may not be that accurate. This is just a toy example demonstrating why this is happening, right? So if you have a NAA molecule, and if you have this spectral line shape and slightly shift variation going on, and if you concatenate and ensemble all of this spectrum, you are looking at a singular value decay of this blue curve, which is very rapid. But if you increase the line shape variation and frequency variation to create a little bit more variation in the individual spectrum, and you're seeing the singular value decay become much, much slower, which means that you will need a much higher subspace model order to capture all these variations. So that motivates us to think about, can we generalize this subspace concept for a more nonlinear subspace or nonlinear manifold concept to allow us to better capture all the practical variations in the real experimental data. So, learning nonlinear low dimensional model from high dimensional data is a very well studied topic in machine learning literature, particularly subspace learning. I think is also extensively studied. And if you look at the literature, and some of them already apply to spectroscopy, and there are all the dimensionality reduction techniques, right, to work very well and identify pattern in the high dimensional data. But as a representation to model the high dimensional variation, so you can incorporate them as a model constraint for data processing and reconstruction, so they not necessarily offer the best representation tool. So, and the advance of deep neural network-based deep representation learning, and really provide a new mean to address this issue, right? One of the most famously used tools is this autoencoder, really allow us to identify nonlinear low dimensional embedding of high dimensional data. So discovering the inherent low dimensional variation of high dimensional data, which particularly fitting for spectroscopy data, which is governing by the variation of small number of parameters. So to do that, again, we can leverage our physics modeling to combining the quantum mechanical prior and some spectral parameter distribution we can learn from experimental data or literature, and we can push them through the biophysical model to generate a large quantity of training data. As we know, to train this deep neural network, we really need large quantity of high quality training data, which turn out to be a luxury for MR spectroscopy imaging, right? But we can generate as many physiologically meaningful data as we like using this physics-driven approach. And we can use them to train this deep neural network. In particular case here is the deep autoencoder. And we can also combine this with some small number of experimental data to fine-tune the network to allow it to better capture population or experimental dependent variation. So here's some example illustrating the superiority of the nonlinear low dimensional model. As you will see, the orange curve, which is the model truncation error or dimensionality reduction error for some test data, consistently fall beneath the truncation dimensionality reduction error from the linear subspace model for the same model order. And here's a particular test case that have not been seen in the training data. If you use a 16 dimension, and the learned nonlinear model really offer you a very good match, almost perfect match with the black curve, which is the ground truth. But the linear subspace model, which is the blue curve here, shows significant modeling bias. And if you want to extend to this multi-dimensional spectroscopy acquisition, you need to tailor the data a little. You need to tailor the network structure a little bit, including some kind of convolution layer to help you further explore both the time and across parametric dimension correlation, such as TE. And because the dimension is much higher, we can't just use the fully connected layer, And then we need to also handle the complex value carefully. So we extend this to a complex value neural network, combining both convolution features and some kind of auto-encoding fully connected layers. So allow us to really identify very powerful nonlinear low dimensional representation. And this test here, we tested for different cases from single T phosphor MRSI, single T proton, and even multi-T proton MRSI. So the learned nonlinear model consistently outperform linear subspace model across different model order. But of course, if you get to very high model order, the subspace seems to be also have some edge, but you normally would not go this high. But I assuming there's some training issue when your linear model gets very high. Okay, so now that you have this model, you can rethink about how you will formulate the image reconstruction problem. So one possible formulation is that you have this physics-driven data consistent, where A basically capture all the spatial spectral encoding prior, and you can add a spatial regularization term that enforce the high dimensional data actually reside in a low dimensional manifold that you learn, right? For example, this kind of regularization penalty. So this allows for very flexible integration of the physics space for modeling and low dimensional prior learned by the deep auto encoder network. And you can solve this using a different kind of algorithm efficiently. But the fundamental challenge here is that if you want to solve this optimization problem, strictly speaking, you have to backpropagate this neural network, a representation network, which can be computationally very demanding. So we actually did this, but then we found that computation time is actually too demanding for very high dimensional 3D data. So we try to kind of reformulate this problem a little bit. So instead of just putting in a spatial regularization, we actually put it in a manifold constraint, and then they enforce that the reconstruction to reside in the range space of this decoder that we pre-learned from the representation network. And then, of course, this allows us to add in additional spatial spectral prior to further enhance the performance. And instead of directly backpropagating the network, we learned a network-based projector to enforce this constraint instead of backpropagating it through every iteration. So this significantly enhances the computational complexity, and also lends itself to efficient algorithms such as projection gradient descent type of algorithm, and still solving this optimization problem very efficiently. So with time limit, I'm not going to go into the detail of the training procedure, but hopefully you get the idea. So here's some simulation result comparing the different method, particularly if you look at the subspace-based reconstruction and then proposed method using a nonlinear model constraint, you can see that we are achieving better reconstruction performance. And this simulation result compare two cases that both use learned nonlinear manifold constraint, but one is directly solving the neural constraint regularization problem, which is the first formulation I talk about. And the second one, which we learned the projection incorporating the efficient projected gradient descent algorithm, we call that RACE. And if you compare these two methods, they achieve similar performance, if not better, for the new formulation. But most importantly, if you look at the computation time, we are accelerating the processing almost by 100-fold. So this is you can achieve even slightly better performance with higher computational efficiency. So this can become more practical for analyzing practical in vivo data. And we tried this on some in vivo phosphor data acquired by Dr. Anki Hennig from UT Southwestern Medical Center. And this is a 9.4 Tesla phosphorus MRSI scan. If you look at the noisy data compared to the denoised data using our neural network constraint, we are seeing much better spatial distribution and a much better spectral structure. And then we are reviewing some, we are better reconstructing some weak metabolite component. And again, compared to the direct formulation and versus our race reformulation, the experiment time is much, much faster compared due to the more efficient computation with similar performance. And it does improve the test-retest reproducibility for not just the major component, but even more for the weak metabolite component, such as the inorganic phosphate, which is an interesting metabolite marker for phosphorus MRSI. And you can do this for proton MRSI as well. And this is the case. We are trying to apply this denoising scheme to a rapid 4-minute acquisition. The resolution is not super high, but as you can see here, compared to the subspace-based reconstruction and denoising reconstruction, the nonlinear manifold constraint reconstruction achieve better spatial pattern and then better line shape reconstruction. Better line shape in the reconstruction really speak to the potential of this physics-driven the model learn integrating physics prior and data-driven machine learning. Yeah, so to quickly sum up these results, so hopefully I demonstrate that physics-driven deep representation learning can be useful in tackling high-dimensional MRSI problem, and hopefully extending what's originally offered by subspace imaging. And this kind of strategy, I believe, could also be useful for other high-dimensional imaging problem where the signal evolution followed by a well-defined biophysical model. And then hopefully I show you that the learned nonlinear low-dimensional model, if you learn it correctly, can really become a more effective and powerful tool than linear models. But of course, we need to have more careful evaluation about the generalization capability because it's nonlinear model typically have this issue. Yeah. With that, I would just like to conclude. I'm hitting the hour limit, and this result would not be possible with amazing students and collaborators, both at Illinois and external collaborators. And this is my group, small but dedicated. And this is where I'm located, Beckman Institute. It's an amazing facility for interdisciplinary research and neuroimaging research. I also like to thank the funding for the research that described in this presentation. With that, I'd like to conclude and happy to take any questions. Thanks.
Thank you so much. It was a very interesting talk. I really appreciate it. So we have one more question from Cyrus. He's asking about the effect of the scanners, basically.
Yeah, that's a great, important question. My hope is that we will get there. There are a lot of practical issues. This goes beyond scientific and technical development. There's a lot of engineering issues to make this quantitative measure consistent. And depending on how you define the quantitative measure, I believe some kind of ratio metric measurement will have more potential in compared to absolute quantification. And then some of T2 and ADC probably could be standardized, but it will take years of effort to get there for sure. So we are just trying to demonstrate this is possible, and we are going to continue to push this direction to make it more robust. As you're probably aware, that in the water imaging community, people are still trying to make T1, T2 measurement consistent across different sites, different scanners. So eventually, I believe we need to get there, but it will take a lot of effort. Hopefully that answered the question.
Vince, do you have a question, or I should ask one more?
No, I was just going to say it's a great. It was actually a really nice talk and a lot of depth. You had a lot of different things you talked about, and Armin and I were looking at it and thinking that a few of the approaches that you're when you're doing the matrix decomposition and the like, we could potentially see some applications of blind source separation type approaches there, which maybe we could chat about if you're not doing already. So
yeah, no, exactly. I think some of those matrix factorization would be very, very useful. Yeah. So MRSI has these unique challenges considering the limited SNR we have, right? But definitely, I think there would be some synergy there.
Yeah, and we've done a little bit of not in this type of work, but we've done a little bit of work with single voxel and multivoxel spectroscopy with blind source separation. So that's something that could be a base to build from. But and I like the fact that you called your group small but mighty, or small but dedicated. I was thinking small but mighty is another term you could use, but yeah. So
thanks.
Yeah. Any other questions from folks? We're right at 2, but we'll take another question if somebody has one.
So question, what about functional MRSI expanding the temporal direction as opposed to spatial? Yeah, this is definitely a very exciting direction, right? So we have been focused on pushing the spatial resolution, and then also kind of pushing the amount of molecular information that you can extract from each individual voxel, because that's so rich, and there's microstructural information beyond the relative abundance that people have been studying, right? So that's what have been focused. So we are not really focusing on functional MRSI, but I do agree that could be a very interesting direction if you zoom, and could be done in a task-specific fashion, right? If you're really interested in a particular molecule dynamic, let's say lactate or glutamate during a functional activation, people have done that for single voxel. I think that we could potentially tailor the scan to less emphasize on the spatial coverage and resolution to the temporal dynamics. And as you increase dimensionality to the problem, and there could be more fun signal processing issue that we could, we could address. But this is certainly a very interesting direction, yeah. But underlying physiological drive, I think to do this, people have done this using single voxel, and what they have observed, you guys probably aware, is that the metabolic change that we have observed follow a different temporal scale compared to the brain activation that we observe in functional MRI. Kind of the metabolic change kind of slower and fall behind the change of brain activation. You need to kind of constantly stimulate the brain for like a few minutes to be able to see the rise of glutamate and maybe lactate. So is it because of the temporal resolution that I have, or is it because there's more underlying physiology that we don't understand? I think all this could be very interesting topic to study, and potentially leveraging some of like electrophysiology and neural interface measurement that people have been developing.
Great. Well, thank you.